Traditionally, the aims of art and science are viewed as being very different, but throughout history, illustration has played a crucial role in conveying scientific knowledge. So please join me, evolutionary biologist and science communicator Dr. Chris Freelance, for this episode of my 5-part series showcasing key examples of where illustration has been key to communicating new knowledge in the natural and medical sciences. Today, we take it as common knowledge that our nervous system is composed of many individual neurons, with these cells communicating with each other electrically or chemically across synapses. The first evidence for this came from the 1800s, from the brilliant illustrations of a brilliant neuroscientist. Santiago Ramon y Cajal was born in Spain in 1852, a rebellious young man who was reportedly briefly imprisoned as an 11-year-old after destroying a neighbour's front gate with a homemade cannon. After spending time studying anatomy in graveyards with his father as a child, he later became an accomplished neuroscientist and pathologist with expertise in the anatomy of the brain. Many of his insights relied on a new technique for studying brain tissue samples. Italian pathologist Camillo Golgi invented a stain called the Golgi method, which randomly associates entire individual neurons with the compound silver chromate, making their morphology visible using a light microscope. It was this staining technique that allowed Ramon y Cajal to start observing neurons from different parts of the brain and peripheral nervous system in unprecedented detail. Using the Golgi staining method, Ramon y Cajal meticulously illustrated the microscopic details of the nervous system like never before. Importantly, his work demonstrated that not all neurons look the same. There were different morphologies for different types of neurons, for example, between what we now know as pyramidal neurons and Purkinje cells. And in different parts of the brain and in the retina of the eye, for example, he noticed distinct layering or organisation of different morphological types. His illustrations were able to even capture the details of the ultrafine dendrites, which we now know end at synapses with other neurons. By virtue of only random neurons being stained, while there were issues with depth of field, as their three-dimensional nature meant not all of the neuron can be in focus under the microscope at once, Ramon y Cajal began to theorise about how it was exactly that the nervous system was structured. At the time, there were two theories in competition. Reticular theory posited that the nervous system was one continuous network, and views down the microscope of dendrites overlapping parts of other neurons seemed consistent with everything being connected. Golgi was himself a strong proponent of this theory. Neuron theory posits that each neuron is an individual cell, and that neurons communicate with each other across what we now know as synapses. And the apparent overlap seen down the microscope simply reflected three-dimensional organisation. With his observations of different morphologies, layers and organisation to back him up, Ramon y Cajal was a strong proponent of this theory. Such was the contribution of the Golgi stain and of Ramon y Cajal's observations and illustrations that in 1906, despite their opposing views, Golgi and Ramon y Cajal shared the Nobel Prize in Physiology or Medicine. With the advent of electron microscopy in the 1950s, almost 2 decades after his death, Ramon y Cajal was finally vindicated. These microscopes allowed it to be clearly seen that neurons were individual cells interconnected by synapses, and this provided conclusive evidence for neuron theory, and finally laid reticular theory to rest once and for all. And now, almost a century on from Ramon y Cajal's death, he is still recognised as the father of modern neuroscience, and a neuroscience research institute in Spain bears his name. And as for his intricate illustrations, which informed his revolutionary view of neuroanatomy, there are museum collections dedicated to preserving the original drawings. And books and posters of these works are still a prized educational resource, and quite simply, rather beautiful to look at.